Hello, welcome to 13 Universal, learning beyond the barriers, learning beyond barriers. And this is Shedro, your biology tutor. We are going to be looking at all topics theory, paper 2. That's AS category, theory in paper 2, series 3. We have done 1, 2, 3. Thank you for following us, okay? Let's dive in. Get your writing pad ready. Get your pen. Let's apply some concepts in biology. Okay, number one, in flowering plants, okay, take a deep breath. In flowering plants, transport of assimilates occurs in phloem tissue between sources and sinks, okay, between sources and sinks. And then it says, explain why a root can be a source and a sink at the same time. Yeah, a root can be a source and a sink. So explain why. Okay, now the first question you should ask is, what is a source? Okay, now a source, why is a root source? A source is where sucrose, all right, or assimilates are either made or provided, right? Where sucrose is made or provided, that's a source, guys. So either it is made or synthesized or provided. Now, can a root be a source? Yes. Okay, it can be a source because it can provide sucrose, right? Especially when the leaf is not matured. In winter, plants shed their leaves. Okay, when they shed their leaves, of course, where do they get sugar from, guys? The leaf is the bakery, okay, bakes sugar, bread for everyone to eat. Now when the bakery is closed down, they will remember the store. Okay, now the store becomes the source. Is that okay? So yes, the root provides, all right, sucrose, all right, and that's by acting as a source, provides sucrose, not just sucrose, other assimilate like amino acid, hormones, and other sugar like glucose, all right, lactose, and or maltose and all of that. Okay, so it acts also as a sink. Sink is where sucrose is stored. Sucrose is stored or utilized. So of course, when the leaves start working, all right, so what happens is that the the roots, okay, start storing sucrose, okay, in form of starch. Okay, so it can act as root, as a source, and as a sink because it provides sucrose, all right, to the other part of the plant when the leaf is not present, of course. So depending on the demand, of course, the year, the period, okay, if there's no leaf, all right, it's going to provide it. So all the starch will hydrolyze and be supplied, broken down to form sucrose. Okay, all right, and then as source, as sink, it stores it. All right, so let's look at what the mass scheme says. So it assimilates as sucrose. Okay, now these are examples of assimilate, that are things that are manufactured. Amino acid, monosaccharides, other name disaccharide, okay, hormones. Okay, I say you should mention it. If you mention to 1 mark, of course, this is how many marks question? It wasn't shown, but this is a 2 mark question. All right, so root as source, the root provides assimilate for other parts of the plant. That's what source does, provides assimilate for other parts of the plant. So reject starch because starch is not transported. Okay, it's only sucrose or assimilate or assimilate. All right, so very, very important. So the root can as well act as a sink. Okay, so what does it mean to act as a sink? That's what the mass scheme says. So the mass scheme says that acts as a sink. All right, root is for storage. Okay, yeah, storage organ, receives, assimilate, to store, grow. Okay, so roots can grow. Remember that assimilate, sink is where assimilates are either stored or used for growth. Okay, now reference to bi-direction transport in phloem. So phloem transport can move from source to sink and sink to source, depending on where the source is and where the sink is. Okay, so that's another reason. Direction of movement changes with demands of plant of named assimilates. All right, so if a particular assimilate is needed by the leaf and is no longer made by the leaf and the root has it, can move from the root. All right, depending on the need. All right, that's 2 marks. Let's go to the next one. In flowering plant, transport assimilate occurs in phloem tissue. Okay, yeah, that's now the same question. So state 2 examples of assimilate that transport in phloem. We've mentioned that. All right, amino acids assimilate sucrose is assimilate hormones. All right, sugars. Look at them. Sucrose and amino acids are assimilates. All right, are molecules that are transported. All right, after they have been synthesized by the leaf. All right, so or by the source. Sucrose and amino acids. Say Maskim says accept other named monosaccharides and disaccharides. Right, hormones like abscisic acid, mineral ions. So the examples: alcohols. Look at organic acid. So write it down if you don't know. Sugar, organic acids, alcohols, proteins, RNA. They are all made in the source, and they can be transported from one part of the plant to another, right, through the phloem. Okay, so just one mark, one of them. Describe and explain the mechanism that is responsible for, this is for, not all, for, typo, for the movement of phloem sap in sieve tubes, right? So they say we should describe. So there are 2 command words here. Describe answers the question how does it go, and explain tells us why does it move. Okay, before we look at the masking, let's try to remember that in biology you must be able to recall the concept you have learned. So this is phloem tube, guys. This is phloem tube. All right, is running from the source. So this is source here. Remember that it has sieve tube. The sieve tube element has sieve tube here. Is that okay? Sieve plate means a sieve plate. Sieve plate they have pores. All right. So they are the ones that are joined together to form a longitudinal tube that forms you know, the sieve of the phloem. All right, phloem tube. Okay, so guys, look at it. You can see that. Yes. So, but don't forget that. Let's assume that this is the sink, right? Sink here. Of course, let's start from the source. This source. All right, source here. This is companion cell. Companion cell helps in loading of sucrose. Okay. This is the source cell, mesophyll source. So this is source cell, source cell. This is companion cell, companion cell. Okay, see, let me call it CC. All right, so at this point here, of course, this is also companion cell here. All right, this is also sink cell. So this is sink cell. I'll call it SC or sink SK. Right? Do you like the name SK? SK International. All right, this is companion cell, right? CC. All right, so let's see how it goes, guys. Are you ready? So let's see how it goes. Okay. Okay, so look at it. The question says describe and explain mechanism that is responsible for, this is for the movement of phloem sap in sieve tube. So before it starts, there are, guys, there are 3 mechanisms that must be discussed here. 3 stages. Number 1 is that we must look at loading of sucrose. Okay, loading is very important. So sucrose must be loaded, you know, but I'm not going to talk about loading because the question doesn't want us to talk about loading. All right, so I'm going to jump that. Or do you want me to talk about loading? So loading, unloading. Okay, so anytime you want to describe this process, loading is very important. I'm going to just mention loading, all right, in passing. Okay, so this is loading and that's unloading, right? Unloading, that's unloading. Okay, unloading, and then we have mass flow. Mass flow, mass flow. So let's go. Are you ready? Okay, but meanwhile, you're supposed to have pause this video and answer this question. All right, let's look at it. So loading is all this process that makes sucrose to be moved by its companion cell into the sieve tube. All right, remember the question says the movement of phloem sap in sieve tubes. So they want us to talk about how it moves from the source here down, all right, to the sink here. All right, that's what we want to talk about. So they want to start from this point, but let me talk about, normally the loading involves companion cell pumps hydrogen ion into the cell wall, all right, and they said hydrogen ions then move back. Of course, hydrogen ion concentration gradient set up in the cell wall of companion cell, then it moves back into the companion cell through a co-transporter using sucrose. So when sucrose gets here, it moves from companion cell into the sieve tube, all right, through plasmodesmatous matter. So sucrose is loaded from companion cell into the sieve tube, all right, by diffusion through plasmodesmatous matter. So when it gets into the tissue, into the sieve tube element here, it then lowers water potential. We have started now. So when sucrose moves into the sieve tube here, it lowers water potential here, and because water potential is lowered there, what happens is that water then moves in from neighboring cells. So water is going to move in from neighboring cell. Okay, so when water moves in by osmosis, what happens is that the volume of cell sap increases. Remember that once volume of cell sap increases here, what has moved in? Why? Remember, we are looking at describe, how, explain, why. So sucrose loaded into the sieve tube element lowers water potential. So water moves in by osmosis, and then the volume of phloem sap increases here. Once it increases, right, once the volume of phloem sap increases, hydrostatic pressure also increases. So in the source there, we have high hydrostatic pressure. So high hydrostatic pressure, I'll call it high hydrostatic pressure, HHP. But I'm taking time to explain this, guys. Okay, so that's what loading causes. Loading sets up high hydrostatic pressure in the source. How? When sucrose moves into the sieve tube element, lowers water potential, and the water moves in, volume increases, and then high hydrostatic pressure is set up. Now at the sink here, sucrose is moving out. So when sucrose is unloaded, so when it moves from the sieve tube here into the companion cell, and then from companion cell to sink cell here, so because sucrose has moved out, the water potential in the source here increases. Sucrose is a solute. When you remove a solute, water potential will increase. So water potential here, guys, watch this, increases. Once it increases, water is also going to move out, because water moves out of the sieve tube, all right, to neighboring cells by osmosis, because there's high water potential there. Why? Sucrose moved out, so that increased water potential. Now because water has moved out, the volume of phloem sap here decreases. Okay, now because it decreases, once volume decreases, hydrostatic pressure will decrease. So we now have low hydrostatic pressure, okay, in the sieve tube here, all right, at the sink. Okay, so unloading sets up low hydrostatic pressure, loading sets up high hydrostatic pressure, and that creates a hydrostatic pressure gradient. So we can now bring mass flow. So therefore, sucrose now moves from the source to the sink down hydrostatic pressure gradient by mass flow. Okay, that is all they wanted to explain here. All right, let's see what mass scheme says. So answer any four. So the first one says assimilate diffuse, all right, into the phloem sieve tube element from companion cell. That's what we're talking about. It moves in here. When it moves in, what happens? Water potential in sieve tube decreases, like I told you, is lowered. Okay, all right, reject water potential gradient. So it's lowered, and then what happens? Number 3, water enters from surrounding tissue by osmosis down water potential gradient. Okay, so that's water enters. So what's the next one? Hydrostatic pressure builds up because increase in volume volume of phloem sap. Water moves in, phloem sap volume increases, hydrostatic pressure, all right, builds up, and then hydrostatic pressure difference, okay, created as accept it, or mass flow happens. Of course, mass flow is happening because of unloading at the sink. The unloading at the sink, movement of sucrose out here lowers, increases water potential. So water is moving out. Is that okay? And then what happens? So water potential gradient, spray potential is gradient setup. Okay, so that's all you need to say in this question, guys. You can replay this video again. So look at this question: tobacco mosaic virus, tobacco mosaic virus (TMV) infects many crop plants. The virus passes between cells in the leaves and can travel throughout plants in the phloem. Let's see what's all about. The enzyme pectin methyl esterase (PME) is involved with production of cell walls. PMV, okay, makes cell walls. The enzyme is also known to influence the movement of tobacco mosaic virus (TMV) through plants. Scientists investigated the effect of this PME, all right, this enzyme, on the transport of TV through plants. So they investigated whether it increases it or decreases. So the scientists used 3 varieties of tobacco plants. 2 varieties, V1 and V2, have small quantities of PMV. That this PMV is that enzyme here. So a third variety has the normal, okay? C has the normal quantity of PMV and was used as control in this investigation. So they want to find out whether, all right, this one that has normal, okay, is a normal quantity that is causing the movement, okay, just to compare. Control is used to compare with other experimental setup. All right, the next one says the plants in each group were infected with tobacco mosaic virus at the same time. The accumulation of the virus particles transported to the leaves at the top of the plant was determined over 336 days. So the result as shown in Fig 5.1. The arrows indicate when all the plants were infected with tobacco mosaic virus. Guys, this is it. Okay, so whenever you're dealing with the graph, all right, check the x-axis. Whatever this is independent variable is affecting what is here. So time or days after infection with TMV is affecting the TMV accumulation in the leaves because it's moving, all right, to the leaves. At the top of the plant, percentage of maximum number of virus particles in control. That's percentage. This percentage, right? So guys, one of the things they're going to ask you here, okay, they'll ask you to compare. If you are asked to compare, right, these 3 of course plants, the 2 varieties V1 and V2 that have small quantities of the enzyme PME, and the 3rd variety C that has normal quantity, they'll ask you to compare. Now guys, if you compare means tell us similarities. Anytime you hear compare, similarities and differences. Okay, let's start with similarities first. Can you see that all of them reached maximum, all right, 100% accumulation? They all reached it. Okay, all of them. Now that's number 1 point. If you are comparing, can you see that they all reached, whether it is C or V1 or V2, they all reach 100% maximum accumulation of the virus. Okay, so we are comparing, we're saying the similarities first. The second similarity is that they all took the same pattern, the same trend. Okay, there's a little delay in all of them, and then there's a very quick steep, so they quickly increase. So this happened here, little delay for this C, for the V, V2, and then there's a steep increase for each of them, very quick increase. Okay, now that's for all of them. Okay, all right, so this is very important. So it does not matter now, the next point, it doesn't matter whether it is small, all right, quantities or normal quantities, they all reached the maximum accumulation percentage. All right, so let's look at the differences. You can see that C reached the maximum accumulation percentage faster than the rest. So if you check how many number of days, okay, each box from here to here is 5 boxes, 5 units. That means 1 box is 1 day. So if you come here, you see that should be like 8 days. So this variety C reached the maximum at 8 days. If you check this variety V1 reached at it, I think it reached at 26 days. Yeah, why this one reached at, because this is 30, that's 35, 36 days. Okay, all right, differences. All right, so what else? You can see that there was much delay for V1 and V2. Okay, for example, the accumulation started for V1 after how many days? From 0 to day 9. That's when it started accumulating. While for V2 from 0 to day 22. Can you see? So while the accumulation started here from right from day 1, okay, for C. So that's, I've compared them. So let's see what math scheme says. Okay, quickly. All right, compare the results for varieties V1 and V2 with control group of plan C. Look at it. Any three. So TMV maximum accumulation is delayed in V1 and V2, like we said. All right, they started with differences. Then you have to quote data to support that. Like we have been quoting data in analysis, you have to quote data. Okay, so V1 does not start to increase until day 9. All right, simply until day 16. So it stops at day 16. All we have seen there. V2 does not start to increase until day 22, until 24. Okay, so these are the things you look at. The features of the graph, where there's steeper curve, all right, less steep. So number 3 says rate at which accumulation increases is lower than the control. If you compare two of them, we have seen that, all right. So ignore reference to time, accept reference to steepness of accumulation in V1, V2 compared to, so you can compare their steepness, the one that is that's steeper. All right, look at this. Number 4, V1 and V2, they all, these are differences, these are similarities. They reach maximum accumulation. So low quantities of PME do not decrease maximum accumulation. So what else? Or normal PME not required to reach. So they all reach it. For any other valid comparison, okay, all are slow to start, then very quickly to reach. So you see all that, all have the same pattern trend. So anytime they say compare, you must state the similarities and the differences. Okay, all right, we're done with that question. Let's go to the next question. The mammalian heart consists of 4 chambers. Explain why the ventricles have thicker walls than atria. Okay, why does the ventricles have thicker walls than atria? I don't know whether you have seen the mammalian heart before. Should I draw it for you? Oh, it's gonna take time, guys. Oh my god, it's gonna take time. Okay, now, all right, just to help you remember it, but pause this video and see whether you can draw this. Okay, pause the video. I'm just trying to sketch it. All right, but I think there's a mistake somewhere. So we can pause the video and work out this. All right, so I have something like this. Yeah, okay. So what is this? So this is gonna go like this. Okay. So this is the center. So I'm just sketching it, okay? Just to help you do revision of this topic quickly, of the heart, human heart. But guess what? Whose heart is this? That's not my heart. Is that your heart? I don't know. All right, guys, just trying to, pardon my drawing. Okay, so I hope you're working on this. All right, so that's it. Just to add something to what we have already. Oh, pardon my diagram. Just quick one. Yeah, can you see that? All right, let's bring this down a bit. This is all right. Yeah, there's another. Okay, do you remember this structure? They are valves. What kind of valves are they? Okay, yeah, okay, good. What kind of valves are they? All right, so we have valves here. Pardon my diagram, guys, just a concept. I want to paint a picture of a concept. So these are 2 valves also. just sketching. These are another two valves. Okay, so guys, watch. You remember, this is pulmonary artery, takes it to the lungs. Pulmonary, this aorta takes it to the body, takes oxygen blood to the body. This is pulmonary vein, brings in oxygenated blood. Okay, this vena cava brings in the oxygenated blood. All right, see, these are the ventricles, these are the atria. Okay, good, atria. So the question says, explain why the ventricles have thicker walls. So you can see the ventricles here. So the ventricles, for example, the right ventricle, this is right ventricle, it pumps blood to the lungs. Okay, and the left ventricle pumps blood to the body through the aorta. All right, so the atria just pump blood to the ventricles. So why do they have thicker walls? Number one, they pump blood through longer distances. They pump blood through longer. The ventricles pump blood through longer distances. Right? So this right ventricle pumps blood, deoxygenated blood to the lung through the pulmonary artery. That's pulmonary circulation. While the left ventricle pumps oxygenated blood through the aorta to the body. That's systemic. So because they pump blood through longer distances, all right, so the blood encounter greater resistance, and because they face greater resistance, the blood requires high pressure, and because it requires high pressure, it will have thicker muscular walls. So you need more muscle fibers on the wall because they pump blood through longer distances. Atria pumps blood through shorter distances. Is that okay? So that is it, guys. So let's see what the mass scheme says. Any two from ventricle pumps blood a greater distance into the systemic, okay, except to the lungs and to the body. So they pump blood, the ventricles pump blood through a greater distance. All right, left into the lungs. So reject left into lungs and right to the body. No, because left ventricle pumps blood to the body. See, okay, when this is contracting, these are atrioventricular. This is seminal valve. These two are seminal valves. This is pulmonary valve. No, this is aortic valve. That's pulmonary valve. Now these valves are atrioventricular valves. This and this. This is the bicuspid. These are the tricuspid. So when ventricles contract, these close, the seminal valves open, blood moves into this. But when the atria are contracting, all right, this open, so blood can move in. Okay, so it moves from left to the body, right ventricle to the lungs. So that's it. All right, ventricles have more cardiac muscles to pump blood at high pressure. So they have more cardiac muscle. Why? Because it says explain. Why? It says reject if smooth or restricted cardiac. Okay, why do they do that? Ventricles pump blood to overcome. Yeah, this is the reason. To overcome high resistance, because they're pumping blood through a longer distance. All right, so guys, that is it. Let's go to the next question. The mammalian heart consists of 4 chambers. I've shown the 4 chambers. Compare table 6.2 to identify the structure in the heart responsible for each function. All right, I think there's a typo error here, but don't worry, the question and the answer are already together. But let me ask you guys, I'll ask a question and write it down. Which structure, all right, is the pacemaker that initiates, stimulates the heart muscles to divide by releasing impulse, electrical impulses or waves of excitation? Which one? Okay, write it down. Which one? The second question I'm going to ask you, write it down. Which structure delays the electrical impulses from the atria from moving into the ventricles, right, momentarily? Which structure? The third one, which structure carries electrical impulse across the wall of ventricles? What is it called? All right, so look at the table here. See, the one that initiated the heartbeat is sinatral node. Delays waves of depolarization between the atria and ventricle is atrioventricular node. Delays it, okay, so that atria and the ventricles do not contract at the same time. Okay, so transmit wave of depolarization, but don't forget that it's not only atrioventricular node that delays it. We have non-conductive tissue between the atria and ventricle. It doesn't allow waves of excitation to get into the ventricles, so that ventricles can receive the field while atria, all right, empties. All right, so the tissue that transmits waves of depolarization through the muscles of the ventricles, they're called Purkinje tissue. Don't forget that. All right, closes when the left ventricle contracts. The valve that closes is the bicuspid valve or mitral valve. I don't want to take you guys. We can watch this video again and then comment. Let's hear what you say. So this is where we stop here. Thank you for watching.